Awesome. All right, so as he said, my name is Julie, and spoiler alert, the title is a total spoiler. I'm going to be explaining to you how the mating system of Erysimum teretifolium turns out to control the distribution of genetic diversity more so than the habitat it occupies. And I'm really excited because there are 2 Erysimum talks in this section, and we kind of got a theme going on here. Anyway, I'm going to start out by preaching to the choir a little bit about how natives are really cool. I think most people here will agree with me when I say we value species, at least partially, because of their intrinsic interestingness. So I'm going to give you a couple of interesting facts about Erysimum teretifolium. First of all, it's an edaphic endemic, restricted to a very narrow range of habitat in the Santa Cruz area. It's restricted to this particular sand parkland habitat, which is a xeric habitat that's island-like in its distribution and separated by mesic redwood forests that we did not originally think pollinators would be able to get through. Additionally, it's a perennial and a hexaploid, which causes some difficulties when it comes to analyzing the genetics of this particular species. When it does flower and produce seeds, it produces siliques that dehisce close to the actual base of the maternal plant, and so it doesn't have any obvious dispersal mechanisms. It does have small wings of less than 1 millimeter, but again, we see most of the seeds dehiscing near the base of the maternal plant. This population, or this species, does have specific restoration goals associated with it, and so we should be concerned with it in that sense. It has only 8 known populations that we were able to find. In a 1998 survey, there were 16 populations, but at least some of those have been extirpated, and many of them are on private land, so we're not able to access them and actually assess them for viability. Additionally, its population numbers range between several thousand individuals to 6 individuals at a single site. And that particular site that has only 6 individuals experienced a recent population bottleneck. It was actually one of the populations that originally had several thousand individuals, but for reasons that are thus far unknown, contracted fairly dramatically recently. Additionally, this particular habitat is threatened by the sand quarrying industry in Santa Cruz, and so for that reason we have more cause to believe that the populations that we can no longer find have in fact been extirpated. And finally, this particular plant represents a first step in analyzing other endangered species. There are multiple other endemics that are restricted to the same area that Erysimum teretifolium is, including the Santa Cruz kangaroo rat and the Mount Herman June beetle. And I know most of you are probably more interested in plants than animals, but this does give us some leverage to tell other agencies why we should be concerned about this particular plant. So our research question was to figure out whether or not there was discernible population structure among occurrences of Erysimum teretifolium. And many of you probably know why this is important. When you have these kinds of instances where species are highly restricted and you want to go about restoring them, restoring extirpated populations, or trying to rescue genetically those populations that are extraordinarily small and on the verge of extirpation, it's important to understand the underlying genetic structure of the populations, mostly for 2 reasons. The first is that you don't want to disrupt any potential local adaptation, and the second is that you often want to maximize genetic diversity in your restored populations in order to make them more stable. So, I'm going to address this using microsatellites. We used 4 microsatellite markers, which encompassed 16 alleles, and for those of you who are interested in how we did this in a hexaploid, we ended up having to analyze them as presence or absence markers instead, and if you want to get into the nitty-gritty of that, I'd be happy to talk to you about it. But it's somewhat tedious, and so I'm not going to go into it in this particular talk. And we analyzed this using 4 different methods. We analyzed it first using the program structure, and for those of you who are not familiar, what structure does is it assesses a group of individuals for genetic clusters, and it decides what the appropriate number of genetic clusters for that group of individuals is, and then it assigns each individual a probability of being associated with a given genetic cluster. So in this example, the individual ID is on the x-axis, and the group assignment probability is on the y-axis. Here we see individuals that are strongly assigned to this red group. In the middle, we have individuals that are mixed, and over here we have individuals that are strongly assigned to the blue group. So I'm going to show you graphs that look like that associated with each population. They're going to contain 6 clusters, so they're a little more complicated, but this simple example should illustrate how the analysis is interpreted. We also performed analysis of molecular variance, and what this does is help us interpret where the variation is partitioned within the species. So we tested this at 3 different levels. First of all, we tested whether variation was partitioned among the groups that were predicted by our structure analysis. Then we tested whether it was partitioned among populations, and we also tested whether it was partitioned within populations. We also used the common measure of population differentiation, FST. So that ranges from 0 to 1, with 0 meaning that 2 populations are virtually identical, and 1 meaning that they're totally differentiated. And finally, we examined isolation by distance, which in this case we derived from FST. And what that does is it regresses genetic divergence against geographic distance and tries to determine whether there's a relationship between the two. So I'm going to present a couple of hypotheses to you that we came into this project with. These are our study sites, the 8 populations that we were able to find. You can see they're located right around the Scotts Valley area in a V, and with this one population that's very outlying comparatively. So our first hypothesis was that there would be no barriers to gene flow. That in fact the redwood forests do not offer a barrier and that genes can travel freely between any and all of these populations. What that looks like in terms of the structure results is that all populations are assigned very similarly. In terms of an analysis of molecular variance, that would show most of the variation partitioned within populations. In terms of FST, there would be no significant FSTs, or they would all be 0, because there's no differentiation between populations under this hypothesis. And finally, the isolation by distance would be 0, because there would be no genetic divergence to partition over a given distance. The second hypothesis was one of partial isolation, that some metric, like the strength of barriers between populations or distance between populations, would control genetic structuring. And so the structure analysis under that hypothesis would look something like this. You would expect isolation by distance to be significant because the strength of the barrier or distance would be a controlling factor. The analysis of molecular variance would find the genetic diversity partitioned between the groups predicted by structure, and the FSTs would be significant between the groups compared between structure groups. And our final hypothesis was that the forests would provide a complete barrier. So under this scenario, every single population would be completely differentiated. All FSTs would be significant. Structure would predict every single one as a different grouping. And we would see under the analysis of molecular variance that the variation was partitioned among populations rather than within them or between the groups predicted by structure. And the isolation by distance would again not be significant because distance would not be the factor, rather the simple fact of a barrier existing would be controlling the genetic divergence. So here are our structure results. There's a lot of colors going on here, so I'll walk you through them. The first thing that pops out to me is these outlying populations, SHGW and Highway 17, which are strongly assigned to a single different group that's not largely shared by these other populations. The next thing is that this other outlying population over here is also strongly assigned to a single color group, which although it shares with this population, it's more strongly assigned than its sister population. And finally, these populations in the core of the species range are largely admixed, especially these 2 in the center, which are almost identical in terms of their structure assignments. So to us, this points towards the partial isolation hypothesis, where this particular species does have some barriers and it's largely controlled by distance, but the barriers are not complete. The analysis of molecular variance tells a rather different story. So the structure analysis that you see in this particular graph is represented with this 12% of variation that's partitioned among structure groups. So what this is telling us is that structure analysis you just saw is only accounting for about 12% of the genetic variation that exists within this species. Most of it, this big 85% number right here, is partitioned within populations, which means that variation is relatively homogeneously partitioned across the species. So that would point to a different hypothesis, which was the first one that I showed you, that there are relatively few barriers acting within this species. For the FST results, we found 18 of 28 comparisons to be significantly greater than 0, which means 18 of 28 population comparisons showed significant differentiation between those 2 pairs. So this fairly well refutes the hypothesis that all populations are uniquely structured and that the barriers are complete, because at least some populations are not seen to be divergent from each other. It also somewhat supports the hypothesis that there's partial isolation, since we're seeing a definite pattern that some populations are different from one another. We also found that all of the comparisons with two of those peripheral populations, the SHGW population and the Highway 17 population that I pointed out to you at the beginning of the structure analysis, were significant. So FST is finding comparable results, that those populations are differentiated. And finally, when we compare populations within the core, so those 4 in the middle that were relatively admixed with each other, we find a much lower FST than when we compare any population to a peripheral population. So like I said before, the higher the FST is, the more differentiated populations are. So we do find significant evidence that those peripheral populations are more differentiated than the ones in the core. Finally, we have the results for isolation by distance. And the results here are, again, a little bit contradictory. So you can see that there is a positive slope happening, so there's some relationship between distance and genetic divergence, but the R-squared is relatively low at only 0.14-ish, and when you do a Mantell test, it's not significant. So this regression is actually not significant, even though it does show a weak pattern. So back to the original research question. Is there discernible population structure among occurrences? We're getting a pretty mixed answer here. On the one hand, the analysis of molecular variants and the isolation by distance analysis is supporting this first hypothesis that there is actually no isolating mechanism among these populations, and that genes can flow relatively freely among them. However, we are getting many FST values that are significant, and we do get some partitioning within our structure analysis that suggests that perhaps the second hypothesis of partial isolation is more accurate for this particular species, but it wasn't as strong as we expected. We came into this experiment thinking that we never see Erysimum teretifolium in the forests, and we don't usually see its pollinators in the forests either. And so since Erysimum is restricted to those sandhill islands, we thought that probably they would be fairly isolated. So what's happening that's preventing this isolation from actually occurring fully and going to the full extent of our third hypothesis, which would be full isolation? We did a parallel study where we were trying to investigate whether or not there was inbreeding depression in Erysimum teretifolium, but we kind of stumbled on this other answer by accident. So you can see in this graph that self-pollinations have significantly fewer seed set than the within population and between population or outcross crosses. So in fact, that's 7 times more between these two groups. So that can potentially indicate 2 things going on. The first is inbreeding depression, which was what we originally thought we were looking for when we did this other study. However, there are a significant number of zeros in the data set for the self-pollinations, and that would tend to lend us towards a different story, which would actually be one of self-incompatibility. Now, it's not known for this species exactly how the mating system works, but in other related species, there's an SRK self-incompatibility system. So for those of you who are not up on your mating systems literature, an SRK self-incompatibility system is like a lock and key mechanism. When 2 individuals come together as putative mates and they try to mate with one another, if they share an allele, most often, as long as the system isn't quote-unquote leaky, that mating will not be successful, and the pollen tube will be arrested at the surface of the stigma. And so we thought, well, maybe that's happening in this particular system. Now, it's highly unlikely, just by virtue of the fact that Erysimum is a hexaploid, or most people think it's highly unlikely, because when a hexaploid goes to try to mate with somebody else, it has 6 alleles that have to not match. And so when you have 6 alleles that have to not match, then you're in this kind of situation that fitness-wise is not so great, especially if you're in a small population, when you have to find an individual that has 6 alleles that are completely different from your 6 alleles. So we thought, okay, maybe this is happening. So we tested for pollen tube growth, and here you can see pretty strikingly the difference between the two. In most of the outcrosses that we tested, the pollen tubes grew successfully. However, in the majority of the self-pollinations, they did not. Caveat to this, we do think that if the SRK system is in place in this species, the system is leaky, because we were able to find self-pollinations that successfully grew pollen tubes. And as you saw in the previous slide, we also had some self-pollinations that did successfully set seeds, But the dramatic number of zeros associated with that data set again pushes us towards the idea that this system probably exists even if it is not the best system or not the most complete system. So back to the original research question. Is there discernible population structure among these occurrences? Ultimately, we came to the answer that yes, there probably is, but not as much as we might expect. So what we think the mechanism for this is that the mating system is preventing individuals that are closely related from mating with one another within the population. Since they have those 6 alleles, it's harder for them to find mates within their own populations and harder for their self-pollinations to be successful. So even though probably the majority of attempted pollinations are self-pollinations or pollinations within a population, they're not the majority of seeds that end up getting set or the majority of seeds that end up germinating or going on to actually produce offspring. So ultimately, that stumbling block of the mating system, or the inability of certain crosses that would be more probable to actually produce seeds, is preventing the kind of isolation that we expected from actually occurring. So we have a couple of management recommendations associated with this species, and it's extremely probable that at least at the Bonnie Doon site, the BE site up here, that I mentioned at the beginning of the talk, which was dramatically reduced from 1,000 individuals to 6 individuals very recently, we'll probably have some kind of reintroduction effort, and there are other sites currently being investigated for potential reintroductions. So we do have a couple of recommendations to put forward in the event that those go through. First of all, we think it's extraordinarily important to conserve the small peripheral populations. Those are the populations where we find the most differentiation and the most potential diversity that could be missed if we allow those peripheral populations to be extirpated. And incidentally, most of those populations are the smallest populations currently, so they do deserve specific conservation attention from that angle, especially when we think about the fact that we do have that 12% of diversity is specifically associated with them. And then finally, we think that reintroductions and rescues should use mixed source seeds. So this is important, especially if the SRK system turns out to be in place, and we are currently trying to investigate identify the alleles associated with that. But if the SRK system is in place, it's extraordinarily important to have a diverse seed source, because if you don't, the reintroduction could fail almost as soon as it starts. If individuals were too closely related and then not able to mate with one another and then not able to set seed, and then the population would collapse again and the reintroduction would fail. So from that perspective, it's really important to use mixed source seeds. And with that, I'd just like to thank all of the people involved in this, including my co-authors and the rest of Team Wallflower, especially Ingrid Parker at UC Santa Cruz for being one of the original instigators of the study related to Erysimum teretifolium, Aaron Tom, Jessica Lucas, Khalid Dijon, the K-Lab, which is my current new lab, for putting up with me working on an old project while in their lab, and the funding, which came from the California Department of Fish and Wildlife and the ALSA Foundation, and I'm contractually obligated to tell you that the NSF GRF is currently supporting me in being alive and feeding myself. With that, I'm open to taking questions.